Lactoferrin is a bioactive molecule that is naturally made inside our body. Lactoferrin is well documented with several health benefits such as immune defense, metal transport, intestinal health, and much more. This nutraceutical has emerged as an important functional food ingredient for dietary supplements and infant formulations worldwide. Recent consumer demand has flooded the global market with various lactoferrin supplements. This film is intended to provide a scientific rationale on how to choose the right lactoferrin supplement for your self-care. Let us start from the basics, source of the lactoferrin protein itself.
Lactoferrin is harvested either from all-natural dairy sources or from genetically modified organisms, known as GMOs. Lactoferrin from GMO or recombinant sources include transgenic cows, yeast genetic constructs, and GMO rice plants. Such GMO-derived lactoferrins, however, are not yet common to the dietary supplement market. Dairy animals provide an abundant natural resource for native lactoferrin, classified in 3 common reserves: colostrum, milk, and whey. Though rich in lactoferrin content, bovine colostrum is not suitable for large-scale protein fractionation due to its limited availability. Also, bovine colostrum poses the risk of innate contamination with blood, other biological secretions, and microbial exposure during the delivery of baby calf. Therefore, milk and whey are the 2 most preferred dairy sources for large-scale extraction of lactoferrin. Let us compare the features of milk and whey as starting materials for lactoferrin extraction. Fresh, unprocessed milk serves as a natural direct source for lactoferrin extraction. Processed whey, a byproduct of cheese manufacturing and a derivative of milk fermentation, is also widely used as an alternative source for lactoferrin extraction. Whey is derived from milk as a byproduct of cheese fermentation through the following 6 steps. Step 1. Starter cultures are added to warm milk. Step 2. After acidification of milk, rennet is added. Step 3. Milk is coagulated to form curd. Step 4. Curd is stirred to release liquid whey. Step 5. Curd is pressed to drain liquid whey. Step 6. Liquid drain is used for whey protein extraction. Comparison between lactoferrins extracted from milk and whey are shown here. Raw milk has neutral pH of about 7, and lactoferrin extracted from this original source is known as milk protein isolate, or milk lactoferrin. Processed whey has an acidic pH below 5. Extraction from whey produces whey protein isolate, or whey lactoferrin. For marketing advantage, however, some companies promote this cheese byproduct as milk-derived lactoferrin. Whey lactoferrins have certain functional limitations compared to milk lactoferrins. To overcome these functional constraints in dietary supplements, whey lactoferrin is generally administered at higher doses compared to milk lactoferrin. Based on these observations, the natural unprocessed milk is the most preferred and superior starting material for lactoferrin extraction. There are several patented advanced methods to isolate and purify lactoferrin protein directly from natural, unprocessed milk. Dairy plants around the world use different procedures to extract lactoferrin from milk. Initial steps of milk fractionation include an ultrafiltration step to remove milk sugar lactose, a solvent extraction step to remove milk fats, a chromatographic step to separate milk proteins, and a salt gradient elution step to isolate lactoferrin fraction. Milk lactoferrin fractions are dialyzed to remove salts and dehydrate the protein solution into a fine powder form, either by spray drying at a hot temperature or freeze drying at a cold temperature. Lactoferrin is a glycoprotein which is easily damaged by heat. Lactoferrin is denatured at 90 degrees Celsius. Therefore, lactoferrin protein powders obtained from freeze drying and spray drying processes are significantly different. Freeze drying process is below -10 degrees Celsius. At this temperature, lactoferrin structure is stable. The freeze-dried protein powder contains no cationic peptide fragments, and shelf life of the ingredient is well preserved. Spray drying, on the other hand, uses heat above 110 degrees Celsius. At this temperature, lactoferrin structure is susceptible to denaturation. The spray drying process generates cationic peptides and compromises the shelf life of the protein powder. Cold storage of raw milk during collection, transport, and storage refrigeration during milk fractionation and protein extraction, and freeze drying below -10 degrees Celsius are vital for harvesting a stable and bioactive milk lactoferrin. Harvesting a pure protein with quality control measures does not necessarily ensure a functionally active lactoferrin. Lactoferrin requires several essential cofactors in specific ratios for biological activity. Molecular activation, or functional optimization, is a prerequisite step before formulating lactoferrin into a supplement. Molecular activation protocols are well established among lactoferrin scientists. A patented functional optimization method, TCR technology, is a proven method to prepare highly potent lactoferrin for food supplement applications. Functional optimization of TCR milk lactoferrin can be verified and compared with other commercial milk, whey, or milk-derived forms of lactoferrin. Bacterial growth inhibition assay. Growth inhibition potential of TCR milk lactoferrin against human enteric pathogen, E. coli, was measured. Bacterial nutrient broth was mixed with TCR milk lactoferrin or regular milk lactoferrin or whey lactoferrin at 1 mg per milliliter concentration. Broth without any lactoferrin served as control. An E. coli strain with green fluorescent protein is inoculated into all 4 broths and incubated at 37 degrees Celsius. After 24 hours, the intensity of green fluorescence in all 4 broths was measured. Note that the intensity of the green fluorescence is directly proportional to bacterial growth density. The test results are displayed. Bacterial growth was affluent as indicated in bright green fluorescence in the control broth after 24 hours. The green intensity was a shade below the control, with regular milk lactoferrin and whey lactoferrin broths suggesting a moderate bacterial growth inhibition. Now, watch the TCR milk lactoferrin broth. E. coli inoculum remained as cell suspension with no growth multiplication whatsoever, a perfect microbial growth inhibition. Cell apoptosis assay. Effect of TCR milk lactoferrin on human intestinal epithelia was tested. Tissue culture medium was mixed with TCR milk lactoferrin or regular milk lactoferrin or whey lactoferrin at 1 millimolar concentration. Medium without lactoferrin served as control. Each of the 4 media were added to cell monolayers. Apoptosis, known as programmed cell death, was examined after 24 hours. View the experimental data. Control cell monolayers showed moderate 5% apoptosis, while lactoferrins from regular milk and whey caused extensive cell death of 35% and 53% respectively. This cytotoxicity is due to high cationic peptides in lactoferrins extracted from regular milk and whey. Conversely, the TCR milk lactoferrin is safe on human intestinal epithelia with negligible apoptosis of less than 1%. These data indicate the protective nature of functionally optimized TCR milk lactoferrin.
Remember, lactoferrin is a bioactive protein naturally present in your body. From a physiological perspective, it is considered a multifunctional bioreplenishment that supports your immunity, intestinal health, and much more. Take-home message: all lactoferrins are not created equal. Therefore, choose before you use. Self-care is the ultimate healthcare.